Hi everybody. I'm really excited about this session because it's really in the spirit of the Mendel Symposium to look at an issue from multidiscipline perspectives. So we're going to be looking at agriculture, food production, biotechnology from the perspective of scientists, right inside doing it, and from the perspective of philosopher ethicist who's on the periphery looking in, okay? So we're going to start with Alan McHughen. I'll introduce him. He'll give us his talk. We'll have some questions after that. And then I'll introduce Paul Thompson, and he'll talk, and then we'll have some questions after that, okay? So first, Alan McHughen. He's, need my glasses. He's a cooperative extension biotechnology specialist, that's a mouthful there, and a molecular geneticist in the Department of Botany and Plant Sciences at UC Riverside. He has a really long-standing interest in biosafety, crop improvement, environmental sustainability. He has even a career-long commitment to public education and scientifically informed policymaking. As a plant geneticist, he has developed commercial crop varieties firsthand, using both conventional and genetic engineering techniques. So really, he's uniquely qualified to bring scientific knowledge to discussions of biosafety and public policies governing food production. And this is a service he has tirelessly provided for over, 30-ish, 20-ish? No, let's not make you that old, right? 20 years. I can't do the math. Dr. McHughen began his academic journey for the undergraduates here as a biology-biochemistry major at Dalhousie University in Halifax. He gives a lot of credit to his undergraduate mentor, G.S. Hicks, who, quote, taught me science and scientific integrity that I continue to use every day, unquote. After earning a PhD in plant science from Oxford and a brief stint at Yale, Dr. McHughen took a faculty position at the University of Saskatchewan, and there his research in plant molecular genetics led to dozens of peer-reviewed publications, book chapters, and conference proceedings, as well as several patents and registered crop varieties. He joined the faculty at UC Riverside in 2002 and has made his home there ever since. At UCR, his work in biotechnology risk assessment, science-based oversight, and agricultural policy and regulation has resulted again in a slew of publications, book chapters, books, conference proceedings, and invited talks, both national and international. So over the course of his career, Dr. McHughen has assisted federal agencies in both the United States and Canada in developing regulations for safety testing and release of GE crops and foods. He served on or reviewed National Academy of Sciences panels investigating the safety and environmental effects of various biotechnologies and their impact on sustainable farming. He's the founding and past president of the International Society for Biosafety Research and a fellow of both the AAAS and the American College of Nutrition. Dr. McHughen has received numerous honors over his career, including a Jefferson Science Fellowship in 2011 to serve as science advisor to the U.S. Department of State, and in 2012 he was asked to serve as senior policy advisor under the Obama administration. So finally, as a gifted public educator and consumer advocate, Dr. McHughen helps non-scientists understand the basics of biotechnology and genetic engineering and the rational assessment of the risk and benefits involved. His book, Pandora's Picnic Basket, The Potential and Hazards of Genetically Modified Foods, received the Book of the Year Award in 2000 from the Canadian Science Writers Association and was, to paraphrase a reviewer, quote, the first book to look at all the issues surrounding these technologies in a clear and dispassionate way. Since then, he has presented lectures and seminars, written popular articles, and done media commentaries, placing biotechnology in the greater context of society and politics. And he stayed true to his conviction that good public policy, whether it's on food production, oil pipelines, global warming, vaccines, or human genomics, requires first and foremost accurate information and the understanding of the scientific foundations. Okay, so with that, let's welcome Dr. McHughen. Thank
you. Well, thank you, Angela, and thank you, Father Ellis, and your committee for inviting me here. This is my first visit to Villanova. I have been to the Philadelphia area in the past, but not to your August campus. I'm very pleased to be here to participate in this Mendel Symposium. Of course, I've been a fan of Mendel since I started my academic career, and even before, when I was an undergraduate, learning about Mendel was instrumental in my choosing the career path that I did. So I'll just, if I have a presentation here, pull that up. Okay, so I'm going to set the foundation for the rest of the symposium. I'm going to talk about the technology, the genetics, starting with Mendel and some of the things that he did, and then going from there, try to bring you up to speed with the state of molecular genetics as it is today, some of the opportunities we have, some of the limitations, some of the concerns that we might want to raise as we developed an informed dialogue over the pros and cons, searching what we should do with this technology and what we perhaps ought not do with the technology. Does this work? Yes, it does. So, starting very quickly, Mendel chose 7 different traits in peas when he was doing his experiments, and I'm sure you've all had some background in the basics of Mendel's experiments back in Brno. The types of traits that Mendel chose to investigate include things like the seed color, the seed shape, whether it's round or wrinkled, the seed coat color, also associated with the flower color, flower position, the stem length, the pod color, and the pod shape. These are important traits for a number of different reasons, and I think that will become apparent as we go along. But if you remember from high school, your high school biology and genetics class, Mendel really was important for setting the foundation in doing his meticulous measurements that other people just weren't prepared to do at that time. Working with the peas and these particular 7 traits, taking measurements of these over several generations, and trying to develop and understand the patterns of inheritance. Right? Why do particular wrinkled seeds or the yellow pod color, how is that transmitted to progeny, and why are the ratios of the progeny not one or the other? And thus developing several of the principles that we now recognize from the father of genetics, including things like the traits. And he didn't know anything about genes, by the way. He certainly knew nothing about DNA. He called them factors, I believe. There were, instrumental were the influences coming from both the mother and the father in the progeny. So, 2 factors are involved in determining each of these 7 traits. One from the mother, one from the father. Sometimes one was dominant, which means it was always expressed. Other times the trait, what we would call now the allele of the gene, would be recessive. And the recessive was there, but it wasn't expressed unless there was no dominant trait or allele present there. So these are fairly basic concepts that continue to work with, or that we geneticists continue to work with today. For these 7 traits, very quickly, the seed shape of smooth over wrinkled, the endosperm color yellow over green, seed coat and the flower color purple dominant over white, pod shape inflated over constricted, pod color green over yellow, flower position axial, that is somewhere in the middle of the plant as opposed to terminal at the end of the stem, and the stem length being tall dominant over short. Now, Mendel didn't know how these things work. right? He was able to notice and record whether a seed was smooth and round or whether it was small and wrinkled, right? That's easy enough to dichotomize and score one as opposed to the other. He wasn't able to ask, well, why is that seed wrinkled? What is it about that recessive gene that makes the seed wrinkled? We can ask that question today. The molecular techniques that have been developed in the 21st century now allow scientists to go in and ask that question that I'm sure went through Mendel's mind, but he had no means to explore it at that time. And only up until recent years have we been able to use the tools of molecular genetics to bring that on board. Now, of these 7 traits, we have several that have now been analyzed at a molecular level, and I'll discuss those in a moment. One of the limitations, though, was that we don't know exactly what varieties Mendel worked with. Okay? The seeds were not saved. So when modern scientists tried to go back to replicate exactly what Mendel did with his crossing and so on, we don't have access to that original pea population. So with some digging, scientists were able to find some of the popular pea varieties, genotypes, that were present in Central Europe at the time, and looked at some of the traits that those have that we still have seed for today. And so there's certain varieties that could well be the same ones, but aren't definitely proven to be the same ones that Mendel worked with. Okay, so as a scientist, a curious scientist, we're wondering what happens when we take one of these pea traits and we eliminate the gene altogether. That is, we know what the dominant trait does, right? It makes a purple flower, for example. In the absence of that, the recessive gene makes a white flower. Well, what happens when we obliterate that gene so there's no dominant and no recessive? I mean, the flower is still there. It's not enough to kill the entire plant. The plant grows, it produces a flower. What color is the flower? What is it that makes that dominant gene produce a purple flower and the recessive gene produce a white flower? And what happens if we don't have either one of those alleles, neither dominant nor recessive? Is the flower green? Is it clear? Purple again, even though there's no dominant? So we can ask that kind of question now. And what we find when we do ask that is that the null effect, and this is, null is the situation where we have neither gene is present, right? Neither allele is present. That the appearance, that is the phenotype of the flower or the seed, is the same as having the recessive allele. So what I learned when I was in high school, learning about Mendel and these dominant and recessive traits, is that the recessive was only active when the dominant wasn't present. And that led me to think, well, that means the dominant gene is actually doing two things, right? First of all, it's making the purple pigment for the flowers. We're taking that flower color trait as an example. It's making somehow making the flower produce the purple pigment. And secondly, it's doing something totally different. It's repressing the recessive allele in the heterozygous plant. Right? Those are two very different things, and it's very unusual in genetics for something to have two totally different functions. And geneticists will look at a situation like that and say, okay, we should be able to split that, right? By mutagenizing, we should be able to get plants that produce the purple pigment but don't inhibit the recessive allele. So then what does the flower look like, right? Is it a light-colored purple? Is it striped purple and white? Or what? Well, we weren't able to do that, but we were able to find out that the recessive allele was always the same as a null. So this leads to the more modern thinking that recessive alleles don't actually work. They're dominant genes that are broken. A mutation in a dominant gene such that it renders the dominant gene non-functional is equivalent to recessive or null. So Mendel, of course, had no way of knowing this and no way of testing it. We can't blame him for not foreseeing this. But he was particularly lucky, or not lucky. That implies it's entirely random. We like to say serendipitous. Right? So perhaps he had some reason for choosing the traits that he did, because these traits are unusual in that they tend to be dichotomous. That is, either the flowers are purple or they're white. Right? They're not some blend of in between. The seeds either were round and smooth or they were small and wrinkled. There was not really an intermediate phenotype evident. Most traits are not like this. So that's why we think that there was some serendipity involved when Mendel chose these particular traits to investigate. When we look at the modern view using modern DNA analysis of Mendel's traits for these four, we still don't know. The other three are still waiting for some enthusiastic student to pick up the trail on those. But these four, we have a couple of Australian scientists a few years ago decided for fun just to track down what happened to Mendel's traits we now know as genes. And we're able, some work has been done since then as well to further characterize what these genes actually do. And as I say, we're not positive that these are the actual genes that Mendel used because we don't have the original genotypes, but they provide the same phenotype. So it's, if it's not exactly the same, it's probably something pretty close. And in any case, it does give us the phenotypes and illustrates the points that we want to investigate. The seed shape, right, which is round and smooth in the dominant condition, is a result of what's called the SBE1 gene, starch branching enzyme. And in the recessive allele, an insertion of 800 DNA bases was stuck into the middle of that gene, rendering it ineffective, right? The gene could not be read and expressed because it has this insert stuck in the middle of the gene. In the absence, then, of the starch branching enzyme, the amylose starch doesn't get converted into amylopectin, the branch starch. This causes a buildup of glucose in the seed, right? During the maturation of the seed, photosynthesis is going on, producing sugars, glucose. That gets transported to the seed. Ordinarily, that glucose gets built into the starch, amylose, and then the starch branching enzyme gives us the amylopectin, and that's what gives the smooth, round pea seed when we have SBE1 functioning properly. In the absence, there's some starch produced, but there's a buildup because the photosynthate still keeps pumping glucose into that seed, right? But it doesn't get incorporated into starch. Doesn't get branched. So there's a buildup there. When you have free sugar like that, it's free glucose, that absorbs water. And during the maturation of the seed, there's plenty of moisture around. The water gets sucked in, it inflates the seed, right? It swells up with the moisture. But then during the drying down phase, as the seed matures, as the plant senesces, the water, the moisture is drawn out of the seed, and the seed collapses, giving it the wrinkled appearance. So there's the explanation that was well beyond Mendel, that we now know explains why the seeds are small and wrinkled when they're in the recessive homozygous condition. There's no SBE1 to make the amylopectin starch. Similar with the seed endosperm color. Stay green is the name of the gene in the dominant condition. It's a bit confusing. It's called the stay green gene. A problem that we geneticists have is that we identify genes often when they malfunction. but we give them the name according to what we see in the phenotype. So it's the mutation that's actually staying green. The normal functioning SGR gene promotes the maturation of the color from green into yellow. Right? When that gene is broken, when it's mutated, then the result is that the seed stays green. It doesn't turn yellow. It doesn't get to mature properly. And the molecular basis is, again, a 6-base insert into that gene, rendering that gene inactive. So the dominant gene, when it's mutated to be inactive, results in the stay green condition. And I'll just diverge a moment here. As you're learning genetics and you get these names, it can be kind of confusing. And one of the most common ones is the BRCA1 and 2 genes in humans, right? Breast cancer, where it's in the news all the time, very common, very scary. Well, my colleague Matt Ridley in Britain, science writer, complains bitterly because so many people, and including geneticists, think that genes are named by what they do, and that we have a whole broad range of human genes that cause disease, like BRCA is a breast cancer gene, right? BRCA1 and 2, they cause breast cancer. In fact, the breast cancer, BRCA1 and 2, when they're normal and functioning, are tumor suppressor genes. They act to prevent the development of the cancers. So they're not there to cause cancer, they're there to prevent cancer. But when they mutate, when it's more obvious to us as scientists and we can see the results, then we call it a breast cancer gene because that's what we see. Okay? Same with the stay green and many other genes that you might encounter as you continue your studies. The stem length trait turns out to be a function of the LE. This is gibberellic acid, or GA3 oxidase, which promotes, it's a plant growth regulator, it's a plant growth hormone. So when it's functional, it causes the plant to grow more than it would otherwise. So the stems grow longer when the gene is functional, when it's in the dominant state. When it's in the recessive state, and this is one of the examples, we're not entirely sure that this is exactly the mutation that Mendel worked with, but it is now a documented mutation where there's a G to A substitution in the DNA in the gene, and what this does is downregulates. That is, the expression is diminished substantially, so there's much less gibberellic acid, there's much less growth resulting from the expression of that gene. It's still expressed a little bit, but not enough to really notice. And if this is the actual gene that Mendel worked with, he would not, in all likelihood, have noticed the slight difference between the tall dominant, where there was full expression, and a substantially shorter, but not completely short, alternative mutated allele. And then finally, of the examples, the seed coat and flower color, the purple coloration, turns out to be an HLH transcription factor, which is not the gene directly responsible for the pigmentation, but a regulatory gene that influences the expression of the flavonoid pigment that gives rise to the purple coloration. Right? Here the mutation, again, was a G to A splice site, which led to a frame shift, giving a stop codon. I know this may be alien to you. Let me put it this way. It just truncates. Everything stops. Right? DNA and the gene expression machinery in a cell, the protein synthetic machinery, is very good at following instructions, but very dumb if something goes wrong. They can't figure out what went wrong as we humans typically would if we see a mistake in a text. We can often figure out from the context what the word is supposed to be, if there's a misspelling or something, but the cell machinery can't. They just stop. And this is what happens with this transcription factor giving rise to the flower color, and so the default is white. There's no flavonoid pigment turning the flower from white into purple. So this is the molecular basis of the 4 traits that we're aware of from Mendel's time. But, as I say, most of the traits that we deal with are non-Mendelian. We do get traits both from the mother and from the father, and the child, in our illustration here, has obviously features from both parents, right? Whether they're horizontal stripes or vertical stripes, the kid has both. Now, this also happens with Mendel's peas, except, of the 4 examples, let me just go back to them for a moment. Oops, wrong way. The downregulated expression. Right? This is not a dichotomy. It's not either tall or short. There is an intermediate stage there that Mendel did not observe, and we don't blame him for that because the distinction could be quite subtle. Lots of things influence the growth of a plant growing in a pot or in a garden. Right? Not only its genes, fertilizer, water, sunlight. So there will always be some variation there. And the likelihood is that even though this was an intermediate phenotype, it was probably not sufficiently great as to rise above the background noise created by these other factors influencing the tall or short stems. So most traits that we deal with are not Mendelian. We still have what we call Mendelian genetics that typically look at the dichotomous traits where you have a dominant and a recessive. But most of the traits that we deal with, whether in agriculture, in human health, in animal studies, are contributory from both parents. Sometimes there's greater influence of one over the other. There are also very few traits that are controlled by a single gene. And again, this was very fortuitous of Mendel, serendipitous of Mendel to choose these because they are single genes. In humans, there's probably there's certainly very few genes, single genes giving rise to noticeable phenotypes. And particularly in medical and health issues, the only one I'm aware of is Huntington's disease. Virtually everything else is a result of multiple genes working in combination. That if Mendel were to look at that in one of his pea plants and with some trait that had multiple genes, he never would have been able to figure that out. So now we move from that background, Mendelian background, into the history of plant and crop breeding, right? It started a long time ago, right? We have 40,000 years approximately of human civilization. Our natural niche is that of nomadic hunters and gatherers, right? That's what our ancestors did 40,000 years ago. More recently, 10,000 years ago, our ancestors started to settle down. They developed agriculture, right? They went to farm communities. They started tilling the soil. That is, they plowed. They displaced all of the species that were in that plot of land previously, the plants, the animals, insects, microbes, everything, pushed them out of the way in order to grow crops. This is probably the most dramatic, undesirable things humans have done to the face of the planet ever. Because when you till the soil, you expose that topsoil, a non-renewable resource, to erosion by rain washing away the topsoil, wind blowing away the topsoil, and that doesn't come back. So around the world, a lot of agricultural areas are suffering by this loss of topsoil that we can't really replace, and this goes back for 10,000 years. All for the sake of growing crops to feed our ancestors in an unnatural activity. It was also the time of the first human-influenced genetic modification. Now, this is not genetic engineering, but it was a genetic modification because as our ancestors came upon a plot of vegetables of some kind, the they'd eat everything, obviously, but they would save some. They would save the best plants, the sweetest fruit, right, or the biggest vegetables that had the most seeds. They'd save the seeds from those to plant next year. Right? And when they finished their nomadic wanderings and came back to it, they would have a larger plot where most of the plants, or many more of the plants at least, had these desirable attributes. They were bigger, they were more productive, they were sweeter, whatever. And continuing to do this year after year meant that the genetic profile of that plot of plants was diminished substantially. Right? Previously, there was a lot of genetic diversity. Right? There were productive plants, not productive plants, vigorous plants, weak plants, green plants, yellow plants, a wide range of genotypes growing in that plot. Our ancestors chose only those plants that were desirable to our ancestors to serve our one species. Right? And over time, the plots became dominated by 1 or 2 or 3 different genotypes instead of the hundreds that were there before. Dramatic loss of genetic diversity, and that continues to plague us today, 10,000 years later. So this plant breeding and animal husbandry developed long before Mendel came along, and Mendel did cite some of this work. Plant breeders were using the concepts that Mendel was able to articulate. Right? Those plant breeders didn't understand what it was. They didn't write it down to try to analyze it. They took advantage of it just from their natural skills, but they didn't really understand the genetic basis of it or try to document it the way Mendel did. So, moving from that, virtually everything we eat has been genetically modified relative to what our ancestors ate, certainly 10,000 years ago, but even a couple of hundred of years ago. So, do we recognize these things? These are progenitors. This is what our ancestors ate. What is it? Anybody?
Corn. Wow.
Right. What about the one in the middle?
Spinach.
Is that it? Yeah. Who said lettuce? Excellent. And finally, one on the right? Okay. So, nothing we eat today, almost nothing we eat today, is in its natural form without any human intervention to modify the genes. Some crops are entirely human or man-made. Triticale, mostly an animal feed, popular mostly in Europe, is a human crop. Right? It's a species. Canola was only developed in the 1970s, very popular vegetable oil now. Right? It's not natural. Many of our crops were developed using mutation breeding, right? Ionizing radiation to scramble the DNA, resulting on rare occasion in useful attributes, useful to humans, not necessarily to the plant. Right? And so many of these, something like 3,000 different crop varieties were developed using mutation breeding to make something useful to humans, and we eat these every day. We don't know about it. They don't cause harm, but the DNA is scrambled. It's unnatural. Now, if you go online and ask about a GMO, this is the controversial stuff, right? And you go to Google and say, "What is a GMO?" You'll get this kind of result coming back, right? It's kind of scary. GMOs are scary. We don't use GMOs scientifically. It is defined in Europe, but it's not defined officially. It's not in our official documents in the US or Canada, regulatory documents, because it really has no meaning. Everything is genetically modified, as I showed you in the previous slide. So we want to be more precise and talk about genetic engineering or recombinant DNA or transgenic. All of those terms are used and are better defined, but GMO is not. Of these 4, the scariest one is probably this in the lower left corner. So what is that, GMO? Sweet potato, right? Well, I can tell you right now that no human has genetically engineered a sweet potato, right? But Mother Nature did. thousands of years ago, using the same bacterial vector that human genetic engineers use now to make genetically engineered or genetically modified crops. That is, Agrobacterium, a wild strain of Agrobacterium, infected sweet potato thousands of years ago and inserted bacterial genes into the genome of the sweet potato, and those bacterial genes, ordinarily Agrobacterium causes a disease called crown gall, and the plant generally doesn't perform well after that. The genes are not transmitted to the progeny. But on this occasion, something happened, and the plants were able to maintain their fertility, they were able to reproduce, and those bacterial genes from Agrobacterium are still in sweet potato today. When you eat a sweet potato, you're eating bacterial genes in that sweet potato. Mother Nature put it there, no human did. So Mother Nature is a genetic engineering agent unto herself. What is a gene? Well, this is where it gets tricky, because I gotta lubricate for this. We all know what a gene is, right? We all know what a gene is. No, we don't. There's no standard definition of a gene. I mean, even I'm a geneticist and I don't know what I do. There are many different definitions out there or characterizations. It's become muddied. When I learned about Mendel back in high school and into undergraduate life, it was pretty straightforward. We knew what a gene was. But it's become muddled as we learn more about DNA and how DNA works or doesn't work. Regulatory sequences, are they genes? Well, they're part of the DNA, they're part of the genome. But in practice, we can say that a gene is a unit of hereditary information carried in a DNA sequence. So a gene informs the cell machinery how to make a particular kind of protein, or alternatively, to express or repress other genes. So regulatory genes, structural genes and regulatory genes are different types of genes, and that's how we use the term in colloquial practice. A gene is like a recipe. It's an instruction composed of the letters A, T, C, and G. If you studied DNA, the structure of DNA, and I'm sure they have, right?
I hope so.
I hope so, right? And the sequence of these bases in order are what provide the information to the cell machinery. Just like, in English we have 26 letters in our alphabet. They make all of the words, the thousands and thousands of words, because of a particular sequence of our letters to make English words. Right? The same thing in DNA, providing information to the cell to make a protein or to repress another gene some distance away. And to illustrate how this works, we'll take a look. Here we have my computer's idea of what a rice plant looks like. If we magnify in on a portion of this rice plant, we'll take the leaf, but it could be from anywhere, and get a magnified view, you see the cell structures. If we look at the middle of a cell, look at the nucleus, we see the chromosomes within the nucleus, and then if we take another step of magnification of these chromosomes, this is where the DNA is residing, right? Vast majority of DNA. There's a little bit of DNA elsewhere. We won't worry about that for now. If we take another closer look at this DNA, we'll see how it's composed in the double helix, right? And composed of these letters A, T, C, and G. If we took all of the DNA out of a single cell in the human body, any cell except a mature erythrocyte, red blood cell, doesn't have DNA. Virtually every other living cell has DNA in it. Right? And it has a complete genome. If we extracted the DNA from any one of those cells and stretched it out, it would be about 6 feet long. So 3.2 billion letter bases long. A lot of information. If we look at this, if we do the same thing with a rice plant, take the DNA out of any single cell, it would be very short. But don't confuse amount of DNA with complexity of organism. Right? Just because we have 6 feet of DNA doesn't make us special. Because then if you do the same exercise with a wheat plant, massive amount of DNA, And I don't think most of us want to, recognize the wheat plant as superior to humans anyway. Not inferior anyway, but the length or the amount of DNA is kind of immaterial to complexity. The average gene is about 1,000 bases long. So the average protein resulting from this is about 300 or so amino acids. If we look at a portion of this, and I know you're not going to be able to read this very well, just take a very small segment of this typical hypothetical gene, and you can see, A, C, T, C, A, G. The words in genetic language are all 3 letters long. So in contrast to English, we can have 3-letter words, but we can have words of many different lengths, very long number of letters. But in DNA, it's always 3 letters long. So we can't read this, but the first word, the first instruction to the cell machinery would be ACT. Doesn't mean anything to us, but it does. The cell machinery recognizes it. We can convert this just as a simple exercise into English. Okay, let me do that. Okay, now we have a simple recipe, portion of a recipe. You may not be able to read this, so I'll read it to you. It says, add 1 cup ice and 1 cup tea. Right? Obviously, we have a recipe for iced tea here. Right? Pretty simple. But we all know how to follow this instruction to have a nice cup of iced tea, right? All of the words are 3 letters long. We understand it. It's only a very small portion. The whole recipe may be a much more elaborate dish that involves a cup of iced tea, but we won't worry about that. This illustrates the point. 3 letters to a word. Every word is understandable. We follow the collection of those words and produce something that we didn't have before, right? A cup of iced tea. In the rice plant, there are 37,544 of these gene recipes. That's a lot of recipes in a recipe book. What genetic engineers, people like me, do is we excise a single recipe, potentially from some other organism, bacterium, another plant. We don't usually use animals. It's conceptually possible, it's technically possible, because DNA is the same in every living thing, right? The cell machinery of everything from bacteria to plant to animal reads the same language. So we could take a gene, a copy of a gene from bacterium, put it into the rice plant, and the machinery of the rice plant would be able to read that recipe the same as it would any of the other 37,544 recipes that it's already familiar with in its own genome. Now, in this simple form of genetic engineering, we slip that recipe somewhere into that encyclopedia of recipes of the rice plant. We don't know where it goes initially. We can't control it. It seems to be random. It's not completely random, but somewhere, when we use agrobacterium, the same mechanism that Mother Nature used to make genetically engineered sweet potatoes, when we do that with the rice, that recipe gets slipped in there, and it looks the same as any other page in the recipe book for the rice plant. Right? Now, we can find out later doing analysis of the genome where it went, but it doesn't seem to be important. The recipe either works or it doesn't work. If it works, it can be expressed at a low level, it can be expressed at a high level or an intermediate level. Right? That's the type of variation we see. We have never seen an inserted gene do something that we didn't predict. Right? It doesn't make a different protein. It doesn't make a different. It doesn't have a different effect on regulating other genes. It either works the way we expect it to in terms of producing a structural protein, or it doesn't work, or it expresses so low as to be useless. And then, of course, the rice plant, like humans, that Mendel would recognize, we have a male set and a female set. So, 37,544 recipes from both parents. Although, in this case, we just did an insertion into, I designated this as male, but it could have been into the female as well, immaterial, because once they cross and breed, the progeny, they will sort out in a normal Mendelian fashion. Okay? And there it stays there. So how do we use this? Well, genetic engineering, we try to make better crops. They could be more productive. They could be more insect or disease resistant. They could be more drought resistant. So in this example, I'll take how we might approach a situation to make corn, which usually draws a lot of water, and water is a diminishing resource, not only here, but around the world. Right? Water is becoming more and more valuable. So if we can grow more of our crops with less water, that would be a good thing. So what we might do is go out into the desert, and I live in California, so it's pretty close. And when I go to the desert, right, I mean, it's not a wasteland. If you've been to the desert, it's not a lush jungle, but there's a lot of plants that do really well there. And you can go out and see a broad range of species of plants that seem to be thriving, even though there's very little moisture. Well, we can look at the DNA of those plants and say, what is it about these plants that allows them to thrive, even though there's not very much water around? And if we're lucky, or serendipitous, like Mendel, and we find there's a gene within that genome for that plant that confers that trait, right? Dominant gene that does something to allow the plant to thrive in the relative absence of moisture. And if we can make a copy of that gene and transfer it over to corn, slip it into the genome of the corn plant, then we might be able to grow corn and turn off the water and still productively growing corn. Right? So corn and soybeans are our number 1 and 2 crops in the U.S. Any moisture saving in growing those crops while maintaining productivity would be really valuable. Everyone would benefit from that, from the farmers to the consumers and to the environment. So, what are some of the genetically engineered products that we have on our market today? Probably the most important one, the first one, was human insulin. So, illustrating again that the gene language is universal. With human insulin, virtually all diabetics are injecting human insulin nowadays. That is a product of the human DNA coding for the insulin gene on chromosome 11. A copy of that was given to microbes, and the microbes were able to read that DNA and produce human insulin. The microbes have no blood. They have no need of insulin. They have no idea what insulin is. But they just blindly follow the instructions, grown in fermentation chambers, produce this insulin, great quantities. It's drawn off from the fermentation tank, purified, put in little bottles, and sold at great cost to diabetics. So that's been on the market since 1982. Right? The first genetically engineered product really that consumers have encountered. We also have chymosin, a lesser known product, but virtually all of our hard cheeses, like cheddar, produced using chymosin, and that was approved by the FDA back in 1990. This is true both in, virtually everywhere around the world. In Europe, 90% of the cheeses there use a genetically engineered product, but it's not regulated as such. In Europe, they decided to exempt that for some reason, and it's not labeled. So European consumers are eating a GMO if they like cheese, and Europeans tend to like cheese, but they're unaware of the fact. We do know Flavor Saver tomato. Everyone's heard of the Flavor Saver. It was really the first whole food product on the market. It was only on the market for a couple of years, taken off. And as of today, there are no genetically engineered tomatoes on the market. If I ask a public audience to give me an example of a genetically engineered food on the market, almost everybody will say tomatoes. Right? There are no genetically engineered tomatoes on the market. We do have some virus-resistant squash, crookneck squash. Right? Not a major product. It's only useful in certain areas where the virus is prevalent. We have virus-resistant papaya. If you go to Hawaii, virtually all of the, well, not virtually all, something like 80 to 90% of the papaya in Hawaii is now genetically engineered to be resistant. And this is also one of the examples of a public sector contribution to genetically engineered crops. This was a consortium of the University of Hawaii and Cornell University putting this together a number of years ago. It's still on the market. It has saved the industry because it was introduced at the same time that the papaya ring spot virus blew into the islands and devastated the papaya crop. So if they didn't have this genetically engineered papaya undergoing tests at the time, Hawaii probably would have lost that market altogether. And then most of the things are things like genetically engineered corn, soy, soybean, canola, cotton, alfalfa, which we don't eat directly, most of us, but we do end up eating the animals that eat the alfalfa. And then sugar beet to produce sugar. So approximately half of the sugar that we eat comes from sugar beets. Virtually all of our sugar beets are genetically engineered. Now, by the time you get sugar, the little packets, it's sucrose, it's purified. There's no DNA, there's no protein, there's no way that you can tell whether that little packet, the sugar in that little packet came from a sugar beet or from the alternative source, sugar cane, right? Genetically, chemically identical. But it did come from a genetically engineered plant originally, and some people get concerned about that. The oil squeezed from corn or soybean or canola, again, trace amounts of DNA and protein, not enough to reliably say that, yes, this bottle of corn oil came from genetically engineered corn plants or conventional corn plants. And then a whole range of genetically engineered drugs, vitamins, and additives. Right? So a wide range of things currently on the market. Whether you call corn oil genetically engineered, I think is a mistake, but as I say, some people are concerned about it. Other crops, the papaya in Hawaii we already mentioned. Genetically engineered soy with healthier oil profile. And this is interesting, right? I mean, we're concerned about oils, vegetable oils. We want to have a healthier diet. So genetic engineering is brought to bear to produce oils with healthier profiles, right? The polyunsaturates. We have golden rice that with increased beta-carotene to fight vitamin A deficiency in poorer parts of the world, right? Just coming onto the market now in Asia. Non-browning, a little bit more frivolous use, non-browning apples, right? The idea here is that parents would give their kids going to school sliced apples instead of a Twinkie. Again, a healthier snack for school. If they do this with conventional apples, of course they get brown and mushy by the time snack time comes in school. Kids don't want to eat them. The Arctic apple stays nice and white and crisp, and the kids find them very tasty. And whether they prefer them over a Twinkie, I guess remains to be seen. The innate potato, another one, a non-browning potato, also lower acrylamide when it's fried. And we all like to eat fried potatoes, but when we fry potatoes, we get acrylamide, which is a known carcinogen. Whether it's a real threat to health, whether there's enough acrylamide there to really affect us, probably depends on how many fried potatoes we consume in a given day. I don't lose sleep over it, but nevertheless, if we can reduce the exposure to carcinogens, that's generally a good thing. And we have new breeding technologies giving us non-GMO genetically engineered products using gene editing or genome editing techniques. These are just coming on stream now, include things like, CRISPR-Cas9, zinc finger technologies, RNA interference, and a couple of others, technical names. But basically, they differ from traditional transgenic or genetic engineering in that they do not rely on the transfer of DNA segments from other species or other organisms. That is, they go straight into the recipe book and change some of the letters already present. So it's much more akin to being a very targeted type of mutation breeding. Right? Mutation breeding does not rely on extraneous DNA or transferring DNA. It uses the inherent DNA and then mixes it up. But the vast majority of products for mutation breeding are deleterious. It's only, rarely once in a while something useful comes up. We can't direct mutation breeding. It's entirely random. But with CRISPR-Cas9 and these other genome editing techniques, we can plan, we can design the CRISPR system to go at a particular recipe within the genome and change in particular ways to give us results that we want to see. And these are new technologies. We're still learning about them, but there will be tremendous growth because it gets around the stigma of transgenics, that some people refuse to buy GMOs because of that. Status of crops in the U.S. U.S. farmers generally very positive, right? Over 90% of 3 of our major crops are genetically engineered varieties. So U.S. farmers willingly choose the GMO varieties. It obviously gives them some benefit. They know that a lot of consumers don't like GMOs, but they grow them anyway. So there must be some benefit to farmers, and so they continue to grow them year after year. They're not compelled to do so. So it's interesting to me that so many people say, yes, I support U.S. farmers, but no, I don't like GMOs. Right? You're undermining your own farmers there that you say that you support. It's an interesting discussion point if you get in with a group of farmers and concerned consumers. So to wrap up, we have Mendel's legacy. It has given us genetic technologies, including GMOs. They give us things like insulin and other life-saving drugs, crops requiring, for example, fewer pesticides, which is important to many people, higher yields and more food, particularly in poorer countries. We don't notice the increase in yield so much in the US or Canada. Disease-resistant Hawaiian papayas that saved the industry there. The safety is a big issue, and we've been studying this since the 1980s, even long before the crops were grown by farmers, looking at various aspects of safety, environmental safety, food safety, health safety, and so on. And to date, there have been over 1,700 independent peer-reviewed safety studies, all of them coming to the same conclusion, being that genetic technologies, GMOs or genetic engineering, presents no new risks. Right? Now, notice the subtle distinction there. That doesn't say there is no risks. There is risks with everything, including traditional or conventional breeding. But genetic engineering presents no additional risks. It's been endorsed by the National Academy of Sciences, the American Medical Association, European Food Safety Authority, the Royal Society of the UK, and something over 200 other professional and medical societies worldwide. And to date, there are no documented cases of harm from the consumption of genetically engineered crops or foods. So that's a pretty good safety record, hard to match. So with that background, and again, this doesn't mean that there will never be problems. We have to be cognizant that there are limitations as well. And we have to recognize that there are risks, but properly manage what the real risks are and not the hypothetical or fantasy risks that many people are afraid of. Bring us back to Mendel and society. I look at this figure from the FAO that we have to produce 70% more food in 30 years' time. That's a lot of food to feed our human population, right? We're going to have something over 9 billion of us on the planet. We have diminishing resources in terms of food, feed, forest, fish. Right? The production of these things requires land, water, predictable climate, and so on. We have to know these things. We don't have additional land. Right? The only additional land we have are reserve lands like the Amazon rainforest. And I don't think very many of us promote the idea of plowing the Amazon rainforest into a soybean farm. The shifting dietary patterns, particularly in China and India, where traditionally a bowl of rice and the occasional bit of fish or vegetables, sometimes meat, that doesn't work anymore. The rising middle class in India and China are demanding high quality protein, animal protein. So much more fish, meat, chicken, fresh vegetables and fruit that they didn't traditionally have, which means there's more demand for the production, worldwide production of these products. the trajectory is catastrophic, right? How are we going to produce 70% more food than we're currently producing? We already have like 1 billion people, 800 million people are undernourished and starving, right? We're already feeding more people than we've ever fed before. So we have this paradox to deal with, and we have more people coming before that population finally levels off sometime in the middle of this century, and hopefully we'll have a sustainable production system by then. Mother Nature has no plan for more than about 6 billion of us. So for those of you who advocate we should go back to Mother Nature, right, back to our hunter-gatherer natural niche, well, you know, it's a very romantic notion, but it's probably not, we're not going to sell that to people, partly because if we do that, we're going to have to get rid of like half the people who are already here. And I don't want to participate in that discussion. Right? Who wants to volunteer? You know, the people that don't want to go forward probably, you know, have already made that decision. Right? The rest of us who are still here, we probably want to stick around. So how do we safely increase the food supply, considering we have fewer land, water, and other resources? We don't want to additionally damage our planet any more than we already have, obviously, right? We've done some terrible things to the planet. It's not sustainable to continue doing that. So we want to have an ethically sound and environmentally sustainable manner. We have to look at every tool available to us, including the wide range of genetic technologies. And this is where the science ends. We scientists can provide society with the technological information. This is what we can do with our genetic technologies. These are the opportunities, these are the potential benefits, and these are the risks. These are the limitations. No, we can't overnight, you know, double the yield of a crop. That is just beyond our technical capabilities. But we can provide that technical information to wider society, to philosophers, to policymakers, to then have an informed discussion as to what we should do. Science tells us what we can do, but I am, as a scientist, I don't want scientists telling us as a broader society what we should do. That is not something I want responsibility for. And I know I'm speaking for my colleagues in the scientific community, but I don't really know anybody who disagrees with that very vehemently. So at that point, I think I will turn it over, and we'll let the discussion progress with a more philosophical and policy side of our discussions this morning. Thank you very much for your attention.